microbial interaction for production of secondary metabolites. I welcome Dr. Boro. Over to you, Dr. Boro.
Thank you.
And then he will ask me, "Madam, आप चले जाइए."
कितना साल आप जान रहे हैं मुझे?
हाँ?
कितना साल जान रहे हैं?
He is my student as well. I was teaching assistant at that time when he was doing his masters. So I used to take classes from him. Everyone. Robin,
Yeah.
continue.
Thank you.
No, I'm not student.
Okay. Good evening, guys. So I will be talking about microbial interaction. Okay. We will see some real life problems and try to solve them. Okay. While trying to solving that, we will see what techniques we can use. Okay. So that you can also use in your say when you do the research. Okay. So we will see what techniques can be done to solve the problems what cancer will get. So you all are probably from biotechnology or other fields also. Other fields. So biotechnology important है ना? सही है ना? So this is important because it means say many new things can be done. Okay. In other fields also you can do, but this is one of the advanced field whereby you can say manipulate the genes, okay, or do many other things. Okay. So when I was a student, I did my MSc here and PhD from India. So when I did my MSc, I say worked with microbes and we tried to get some paper. Can I get some new products? Okay. So our aim should be said that we should produce something new, or say we should get some new products, say new enzyme, new say antibiotics. Mera naam bhi hoga, I will get the name, fame, money also I can, isn't it? So that, and during my student life, say if I get some paper, I will say I will be placed in some other university, isn't it? So paper is important. For our line, we should get some good work, which we need to good paper. Okay, in some good journal, which will say give me some good job like that. Okay, so we are now also running for good papers because for our promotion. Okay, so these are the things. So now, say antibiotic was this first antibiotic was discovered. Which was the first antibiotics? Penicillin. It was discovered by. So how he discovered? By chance. So it was growing some bacteria, by chance one mold came and it was here. So now if I need to say extract some antibiotic, new antibiotic, okay, if I want to extract some enzyme or antibiotics from the soil or say from say microbes, can I get some new antibiotic? What is the possibility? Possibility is very less. Okay, to find a new microbe or a new antibiotic, because not many people have been doing this. Okay, they have been extracting the soil or the trees anywhere, and they are trying to say extract the microbes and say antibiotics. But for the last say 20 years, no new major antibiotics have been discovered by that way. Okay, only two antibiotic was discovered in 2010. Okay. They modify some process. Otherwise, when you extract the microbes from the soil, same microbes will come. So you won't be getting any new microbes, new antibiotics. Okay, same microbes, same antibiotics. So what they did, what this scientists they did, they took that also. When you say take the say soil, there are bacteria which can be cultured in the clay. Okay. There are many bacteria which cannot be cultured in the clay. Okay, there are many bacteria. So what these scientists did, they took the soil and say they kept that soil solution and they grew that bacteria in there only, and they evaluated their potential to produce microbes. And by that way, they could produce that multiply that microbe. They cannot be cultured in media. Okay, they can be cultured in that native soil where they are present. Okay, they cannot be cultured in media. Okay, so by that way they produce. Okay. So as such, when I am talking to you, we are interacting with one another, is it not? So in the life, in the soil also, microbes are talking to each other. They are also working, say talking to each other. Say some are say fighting with one another because I need this space. Okay, they will kick out, they will kill that one, they will kill by using some antibiotic. Okay, they will produce some antibiotic, some secondary metabolite to kill them, so that I can eat that food. Okay, and some you also give, I also give, mutualistic. Okay, so they are both ways. Okay, they are killing also, they are say mutualistic also. So we'll see what things we can do like that. Okay, so my talk is microbial interaction for production of secondary metabolite. Okay, how they interact. So this also produces some new metabolites or antibiotics, because if I am alone, you are alone, we won't talk, we won't do anything. But when we are now side by side, I will do something. Okay, I need to kill it, so I will produce something that may not be produced when they are alone. एक माइक्रोब जब अकेला रहेगा वो नहीं बनाएगा, उसकी जरूरत नहीं है. Okay, they are by doing, they can live say happily. When they come together, now competition is there. Now I need to kill it. Okay, then only they will produce some antibiotics or say products. Okay, so this is very important to produce the secondary metabolites or antibiotic therapy. These are all secondary. Okay, so how we did? When we were culturing some microbes, we got this plate. Okay, this plate we got. Okay, red color plate. Some fungal mycelia was there, and some red color spots were there. Okay, you can see that. Okay, this is the fungal mycelia with some red parts. Okay, now I gave this topic to one of my students. Okay, just go and do something. Okay, is it a true fungus or have bacteria or what? Why the color is red? Okay, so we searched for literature and we found some of the same endosymbiotic bacteria are there. Okay, some bacteria which lives inside the fungus. Okay, endosymbiotic bacteria, they can live in the say animals, humans, plant tissues, microbes also. Okay, so some bacteria are there which can live inside the fungus. Okay, so they are also present. Okay, one of the very good example is your rhizoxine. Okay, it's a say anybody name? Alcohol. Okay, rhizoxine 1-alcohol is produced when this rhizopus, it's a fungus, interacts with the rice. Okay, the rice and seedling blight. It was a seedling blight. Okay, fungus, rhizopus is there. Okay, when it enters, it will cause one disease, seedling blight. Okay, and then they will give. Okay, say they will give the plant. And say they in that interacting plants, one alcohol was produced, which is your rhizoxine. Okay, rhizoxine is there. Okay. So when the scientists studied deeply, they found out that this fungus is not producing the rhizoxine. Okay, fungus is not producing the rhizoxine. It is producing only when one bacteria is there. Okay, when one bacteria is in association with this fungus. Okay, rhizopus. That bacteria is the Boro. This bacteria, when it is present with this fungus, then only they are producing rhizoxine. So they are endophilic symbiotic bacteria. So these also we thought that some fungus will be there. Okay, some fungus will be there in association with the bacteria. But we don't know if it's a endosymbiotic bacteria or they are just saying this is fungus, this is bacteria. They are living together or they are living inside. I don't know. So we try to evaluate that. Okay, so for that, first of all, what we should do, we should identify the fungus, we should identify the bacteria. What are they? If they are present, the bacteria kaun sa hai or fungus kaun sa hai? Humko identify karna padega. Okay, so we identify which is part. So we try to isolate. For that, we need to isolate the fungal and bacterial cultures. Okay, and then spin the fungus and spin if the bacteria is present in that culture or not. Okay, and then if they are there, isolate that one and then do the characterization of the bacteria. Okay. So we view that fungal culture in media. Okay, in media, and by providing antibiotics, what we can do, we can kill the bacteria. So in this cultures, you can see they are devoid of this red color one. Okay, here the association is there. Here the association is there, but in here these cultures are not having the red culture. This bacteria, okay, they are having the antibiotics. Okay, so they kill the bacteria wherever only the fungus is there. Okay, only the fungus is there. Okay, so we could purify the fungal fungus. Now we will purify the bacteria also from there. So now what we will do, we have to identify the fungus and the bacteria. what we did, we isolated the DNA. Okay. আমি এই প্রশ্ন নিকালে DNA চলাইছিল Okay, so we'll do the isolation of DNA and we'll characterize it. We'll try to identify what bacteria or what fungus is there. Okay. So we'll do that, and it was done by using 16S RNA amplification. Okay, so you know that there are ribosomes in our body or in microbes, and what ribosomes are there in the bacteria and what ribosomes are there in the plant. That's the so these ribosomes, they have some large subunit and small subunit. So that small subunit has 16S part. Okay, one 16S part is there. So this 16S will be amplified. They are conserved. They have same sequence all throughout their history. Okay, so they are same because they are making the protein. They are the protein making machinery. Is it not? So they make the protein. So whenever there is mutation, they will die out. Okay, so they are conserved. So now, if we sequence that one, that 16S part of that ribosomes, we can say that okay, this is this bacteria because this is not mutated. It's a very less mutation is there. Okay, they are same throughout their lifespan. If it is E. coli, this is E. coli. If this is Bacillus, this is Bacillus. No changes will be there. Okay, so we will sequence. We isolated DNA and we did the PCR amplification using the primers for 16S RNA. Okay, when we did that, so you can see that they have two amplifications. That is for SS7 that was having the bacteria. Okay, and this is a positive control for E. coli. Okay, so we kept a positive control E. coli so that we can know that my amplification is correct or not. Okay, so we got two amplification. This we can say that okay, this is amplification product. Now we send it for sequencing to know what bacteria is this one. Okay, so now we can do the microscopy also. Okay, so bacteria, which bacteria is that? We will do the sequencing. Also, we will know what is how the cell looks like. Okay, what are the characteristics? So we will do the fungal hyphae staining with lactophenol. Okay, lactophenol cotton blue, and then see what bacteria is this one. And then fluorescent microscopy can also be done. Okay, fluorescent microscopy also. So this is my fungal culture. Okay, so this is my control fungal culture, which is devoid of your bacteria. Okay, it was treated with streptomycin. Okay. So this bacteria, now this fungal culture is having the bacteria. Okay, because the bacteria was red in color. Okay, now you can see through your microscope. You need not to use any staining method, just red color is the bacteria inside. Okay, so the bacteria is inside. Now we'll do this fluorescent staining. Okay, so let's see next one. Yeah, this one. This is we have used fluorescent dye. Okay, Cyto 7. So we did the staining. So when we did the staining, it is the fungal culture, and these are the bacteria that are fluorescing. Okay, so we did the fluorescent microscope. So what we did, first of all, we did the 16S sequencing, then we did the normal microscopy, how that our cultures look like, and now we did the fluorescent microscopy. Okay, so we did this is the only bacteria, this is the bacteria having the inside that fungal mycelium. Okay, so they look like it. Now we'll try to see if I can say move this bacteria inside that fungal culture. So what we did, we took the plate, okay, PDA plate. This is a trans, एक गड्ढा लगा दिया इसमें. This side we grew the fungus, and this side we grew the bacteria. Now the mycelia, when they will move through this trans. Okay, ये इसके ऊपर से चला जाएगा. This is one. Okay, ऐसे जब move करेगा, and this tips will be there. The bacteria is growing like that. They will touch and they will try to move inside. So this is how we do this one. Okay, so they are moving like this. Okay, this one. They are moving like this. Okay, they are not over this one. They are inside this one. Our main aim was that is it present ऊपर में है या अंदर में है. So how we prove? We did that fencing. Okay, this one. So fungus is growing this way. Bacteria is there. So when we did So some fungal mycelia will be growing like this. So it will touch the bacteria and they will go inside that one. Okay. So this is how we showed that. Okay, this is inside one. By fluorescence microscopy also we did. Okay. This is also one way to show that it's going inside. Okay. Now we'll do some preliminary work like some biochemical tests. Okay. Some morphological tests. Some tests are there. Okay. And then we'll do another test which is known as your FAM, fatty acid methyl esterases, to identify the bacteria. So this is also one of the methods by which you can identify. This is bacteria. What bacteria is this one? Okay, FAM, fatty acid methyl esterase, because every bacterial cell has different fatty acids over their surface. Okay, if I take the E. coli, if I take the bacillus or other things, they will be having different fatty acids over their surface. So if I check the fatty acids, okay, this is done in GC, okay, GCMS. So if I do the fatty acid analysis of that one, this profile will be different from this one. So library is there in the machine, so I can compare with that and then say that this is this bacteria or this is this bacteria. Okay. So we did the fatty acid methyl esterases test for this one, and other ticks are there. Okay. So this is how my cells look like. Okay, this is the pure culture of the isolates. And this is how in SAM it looks like. Okay, scanning electron microscopy. It will give the topography. Okay, topography. This is rod shaped, how this look like. Then ground staining. Okay, and this is the morphology: round, smooth, red color, rod shaped, drum negated, approximately what is the weight length. You need to describe this thing. Okay, so we did this. And other biochemical tests can it use some say. Sucrose utilization like fructose use karta hai ki nahi, other things. Okay, another test. So this is antibiotic susceptibility test. So every bacteria has some antibiotic susceptibility. So what is the antibiotic susceptibility of the bacteria? So we did this also. Okay, so you need to have this background. Okay, now say nowadays we can directly sequence the bacteria because the cost is very less. Okay, because the cost is very less, you can sequence. Otherwise, say if I need to identify a bacteria. If I need to identify bacteria, I need to use different techniques, not one technique. Okay. Say I need to do the cystinase, I need to see the cells, I need to do the biochemical test. Only one test cannot be reliable. Okay. No one test can be reliable. Okay. If I do the sequencing also, you need to do the biochemical test for the other. Because one method won't be reliable. Okay. So this is known as the polyphasic. Okay. Other type of data, which is known as polyphasic, means all the methods, at least 3-4 methods, have to be combined together to identify. Okay. You cannot rely on cystinase, you cannot rely on the microscope, you cannot rely on anything. Okay. You need to club the information together, then only you can say that okay, this is this bacteria. Okay. So this is known as the polyphasic approach. Okay. Together, when you combine all the methods, that is known as the polyphasic approach. So we need to do all this biochemical, antibiotic tests, everything. Okay. So this is your FAM, fatty acid methyl esterase. So what we did, we extracted the fatty acid from the bacteria and we did the DCMS. Okay, when you give the DCMS, so they will give something and they will what do you say? Compare with the standard that thing is in their library, and they will say that this is this bacteria. Okay, so they say that this is. Okay, so now this is the fungal culture. This is how the fungal culture look like. Okay, and we did the sequencing. This one is the bacterial genomic DNA. Okay, and then PCR application of the cystinase rRNA, and we did the sequencing. Okay, so the last result of the bacteria, it came out to be Serratia marcescens. Okay, the bacteria is known as your Serratia marcescens, and this is through your GC. FAM analysis, 16S, all the things collaborated say that okay, this is Serratia. Okay, so एक भी कोई भी दूसरा नहीं बताया. Okay, all the results corrected that okay, this is Serratia. Okay, and then release the now we can know that okay, Serratia. If we search the literature, we can say okay, Serratia is producing the red color pigment. Okay, now we know the pigment also. Okay, producing. So we can confirm this. So this is Serratia marcescens. Okay, through all the techniques. Okay. So it was 96% similar to the that is in the library. Okay. and we did the fungal sequencing also, and it was coming to be mucorurines. Okay, so it is also 96% similar to that of the gene bank database. Okay, now suppose I have sequenced the my bacteria, okay, and I will compare with that database in the gene bank. Okay, so when comparing, it should be minimum of 95% similarity. Otherwise, you cannot rely. If it's say 94, 93, 92, it can come. It is not reliable. Okay. More than 95%, then only you will take that this may be this may be this, and you have to compare with other things. Okay. So this is now my sequence. My fungus is now your mucor irregular, okay, and bacteria is your Serratia marcescens. They produce a red color pigment, and they are both pathogenic. Serratia is also pathogenic to humans, and mucor is also pathogenic to humans. Okay, so this is the parasitic tree, Serratia marcescens. Okay, so this was compared with the other bacteria that are there in the library. Okay, these are all Serratia. Okay, and this is mucor irregulars. Okay, now can you see some this ATCC, DSM like that? Can you say what is this one? ACTC, ATCC. Yeah, ATCC is the full form. American Type Culture Collection. Okay, so whenever if I say isolate a new organism, I will say this is new organism. Suppose this is a bacteria I isolated. Okay, this is E. coli only, but I said that hey, this is Robin E. coli. Okay, so can you accept it? Can you accept it? So what the scientists did. So if I say isolate a microbe, I need to submit some gene bank. Okay, and my saying won't be say it's a Robin coli. Okay, it is E. coli only. Okay, so I need to submit it to some gene bank. Okay, some microbial culture collection center. Whereby they will keep it, sequence it, and identify it, and they will say that okay, this is this. Okay, your saying is not valid. They will say, and you have to keep that. Okay, so one is your ATCC, American Type Culture Collection, which keeps and identifies it, and they will supply this bacteria whenever you need, or anybody of you can buy okay, from their center. Okay, in India also we have two centers like that. To keep, say you can deposit this culture. Okay, one is your MTCC, Microbial Type Culture Collection, that is in Chandra, UK. Okay, another is your NCCS Pune, which is known as your Microbial Collection Center, MCC, APCC, MPCC. Okay, and KTCC also is there. I don't know which is from what, and DSN is from Germany, some Microbial Collection Center. Okay. So these are all collection centers. Whenever you isolate a microbe, you have to deposit there. Okay, at least 2-3 deposit centers. Then only it is valid. Otherwise, not valid. Okay. Now we'll see the microbes. So I said that my bacteria, Serratia marcescens, they goes inside the bacteria. That is your mucor irregulars. Does it? These are all exclusive, or they can go inside any of the fungus. So we'll check that. Okay, so this bacteria, does it go only inside the mucor, or it can go inside the other bacteria or fungus also? So we'll check that, and we took some fungal pathogens like Aspergillus flavus, Solani, Physarum, whichever is present in our lab, and checked it. Okay, this is what it looks like. So in Aspergillus, it doesn't go. So what we did, we cultured the fungus in the middle, and we put that bacteria in the forequarter. Okay, so if if it's interacting, it will go inside. So here you can see that it's not interacting. The fungus has overgrown. Here also it's not interacting. Here also it's not interacting because they have bypassed it. Okay, here somewhat is happening. Okay, something is happening. Here also say it has stopped the growth here, but otherwise it's growing. Okay, it's not growing inside. This is the mucor irregulars which we took. Here it's going inside. Okay, and here it's the red one. Okay, something is being done here, but these are not interacting. Okay, so among these, only this and these are interacting. These four are not interacting. Okay, so we're checking whether it goes inside or interacting with the other fungus or also. So other mushroom samples also we took. They are not interacting with other mushroom samples. Okay. Only one that mucor and the fusarium of this body. Okay. Otherwise, they are not interacting with the other one. So this is again we did the fluorescence microscopy if it's inside the fungus. Okay. So we just this is mucorales, my bacteria, my fungus, and they are inside. In other control bacteria, in other this one, they are not. Aspergillus, they are not going inside. Okay. This is also they are not going inside. Okay. In case of fusarium, they are interacting. Is it not? Fusarium may be so fusarium, they are killing the fusarium. Okay. They are not going inside. They are killing the fungus. So this have the antagonistic effect. Okay. So we did the Biochemical Test and we saw that actually this bacteria is killing the fungus. Okay. This bacteria is killing the fungus. It's not going inside, it is killing the fungus. Okay. So Serratia muscellans showed antagonistics against fusarium of this body, okay, by doing the plate culture. Now we will try to isolate the metabolites from that one. Okay, so mera ek bacteria hai, I have one bacteria, I have the fungus. So they will produce some metabolites, this will also produce some metabolites. When they come together now, they are producing some new metabolites, new chemicals, we will see that. Okay. New metabolites. So this is also producing, this is also producing. When they are now together, are they producing different something? Okay, so we'll do that. So detection, isolation, and quantification of secondary metabolites produced during this bacterial fungal interaction. Okay. So whatever course that has been taught in your biology, all will come one by one. Okay. So we have identified the bacteria. What techniques you do in microbiology, we did. Okay. Now we will isolate the secondary metabolite. Okay. So for that, what we did, we need to grow the bacteria. So at what temperature, what pH my bacteria goes, what pH and temperature my fungus goes. We have to optimize it. Whenever we need to produce the same metabolites, we need to first optimize what temperature and pH is required for the growth of the bacteria and the fungus. So we say optimize that, and we optimize that. Say, and we found that this is the highest growth is observed at 25 to 35 degrees Celsius at pH 5. Okay, this is the optimum growth. These are not helping it out. Okay, and this is the optimum pH that it is going. Okay, 5. Okay, so this is your Serratia muscellans. So at what temperature, and what pH your metabolite is being produced, that also we optimize. Okay, so it was found that maximum pigment, that red color pigment, this is red color bacteria. Okay, so red color bacteria is being produced from 25 to 30 degrees Celsius at pH 5 to 6. Below that is small, above and below both are not good. Okay, so above 30 degrees Celsius, it inhibits. Bacteria is growing. Okay, so bacteria is growing, but it will inhibit the production of that red color pigment. So we need the red color pigment. Okay, so by this way we say that high temperatures above 30 degrees Celsius inhibit the pigment production. Okay, and acidic pH stabilizes the pigment. So acidic pH is also required for us. Okay, so this 5 to 6 pH is required. So now we will extract that from the bacteria. Okay, we will extraction of the bacterial pigment, then purification in TLC, in linear chromatography, and then we will do the spectroscopic analysis. Okay, so when we extracted that one and we did the TLC here, you can say this is the crude extract. There are many pans. Okay, now we'll take out this portion, and then run again. This is the purified fraction. Okay, only one fraction will form. Otherwise, there are many fractions which we don't require now. Okay, so we purify that, and we did the UV spectroscopy. This is the UV spectroscopy. So when we scan that from 300 to 700, okay, this is the purified one, red one, and this is the good one. You can see some bumps are here. So these are now clear. You can see this is just pure pigment. Okay, so this is purified. So we did the PLC to purify that. Now we did the UV spectroscopy to see what its peak. Okay, so literature wise, we हमको मिल जाएगा which peak is this one. Okay, it's a production. So now what we will do, we will identify that. Okay, so we will identify by using LCMs. That will give, okay, this is this compound. They will directly say that this is this compound. Okay, so we will give the LCM. So we again purify, dissolve in methanol, and then purified using some filters and did the LCMS. Okay. So and then this will give some peaks which will be the MS of that, and this will say what compound is this one. Okay. So this is the first one is your crude extract. Okay. This is the crude extract. We didn't purify it. Next one is the PLC purified one, and third one is the standard which we bought from the company. Okay. We know till now we know that this is producing. So from the Sigma we bought this one. Okay. So when we did the LCMS, they give this peak and this peak. This is the purified one. So we get the less number of peaks here. Say lot many peaks. Okay. And this is the purified one. So one peak is there. So you can see this this one site is your 4.44, 4.5, and 4.6. Okay. When you did the MS of this one, okay, LCMS. Okay. So when you did the MS, it is coming to the same point. Okay. 324.4. Okay. So this is saying that okay, this is producing. The compound is producing, and this producing compound is required in many of the research, like in cancer treatment also they are using, and they are saying both good and bad effects are there. Producing. Okay. So this is how we identify that this is. So now what we'll check its properties. Okay. We'll check the antimicrobial properties of producing. Okay. Does this producing have antimicrobial properties? They have, because they can kill the bacteria. Okay. So when you check this against some bacteria like Bacillus subtilis, Bacillus cereacea, Staphylococcus, E. coli, and Xanthomonas, they are also killing the bacteria. Okay. More Staphylococcus aureus is being killed, less is your E. coli. Okay. E. coli is gram positive or gram negative? Huh? Gram negative. So gram negative are difficult to kill. Why? You have both gram positive and gram negative bacteria. Okay. Gram negative are difficult to kill. As compared to your positive one, gram negative are difficult to kill. Why? Yes. That is also one. Any other one? Because they have blocked many channels. Okay. If the antibody also goes inside, they will be excluded from their body. Okay. They go inside, but they are excluded out of the body. So they have blocked many channels over their surfaces. So even if the antibody enters their body, they are expelled out from them. So they have blocked many channels. Positive don't have. So they die. Okay. So E. coli is somewhat means gram negative are somewhat more resistant. Okay. So highest activity was against Staphylococcus aureus, lowest is in case of your E. coli. Okay. And we also check the this antimicrobial properties against some of the fungus. Okay, that's mucor, then Fusarium oxysporium. It kills the Fusarium oxysporium. Okay, it's called marvel. Otherwise, others are not safe. So this is how again we show that how it hits. Okay, how it enters. It enters by forming some pores in the fungal cell wall. Okay, in the cell wall, what they do, they will form some pores. Okay, this producing actually are involved in producing some pores in the fungal. So suppose this is the fungal hyphae. Okay, how do you enter? They will expel out that producing. This producing makes some holes in the acid, and they enter like that. Okay, so they create some holes. In case of your Fusarium oxysporium, when they created some holes, they eventually die. But in case of mine, they entered and they multiply very fast, so they cannot be killed. But Fusariums, they multiply, they die. Okay. So this is how we show that this produces some pores. So this is the control one. Okay, so they don't have the red color one because it doesn't enter. My this is propodium. This doesn't enter. Okay, because they don't have the pore. Okay. Now when we apply some producers, purified, not the bacteria. Okay, purified producers when I apply, they have the red color. The bacteria is not there. Why the red color has entered inside? Because there are some pores. Here you don't get the color because there are no pores. Here producing is forming some pores. Therefore, this dye has entered propodium iodide. Okay, we have increased the producing, so it should increase the pore formation. So more color is there. So we show that by applying producing, it say forms pores, and so that my bacteria enters. Okay. Oh, so this is one. And now we'll see if, say, my bacteria, my fungus is producing some secondary metabolite. If they come together, they produces some new antibiotics or say secondary metabolite or not. Say we again extracted the secondary metabolite, then purified it, and then did the LCMS. Okay, and they gave lot many things like this. Okay, this is the pure bacteria. This metabolite. This is the fungal metabolite. Okay, this is the interacting metabolite. Now they are common. These are common. This may be common. This may be common. Are there some unique one that we will find? Okay, so unique one will be say produced during the interaction only. Okay. So these are we used it from say all our data, metabolite data. So now we can say that this one is only produced during the interaction. This one only produced in interaction. This one. Okay. This is produced only in the fungus, and this is only in the this bacteria, this everything. Okay, so we are interested only in these type of things. Okay, which is produced only during interaction. Okay, so we will see that. So 11 compounds were produced only in the pure bacterial culture. Okay, 22 compounds are produced only in the fungal culture, and 13 compounds are produced during the bacterial fungal interaction. Okay, so after reducing all these things, we can now say that 13 compounds are produced during bacterial fungal, which are now we are interested to verify and say what compounds it made. Okay. Yes, you yeah, compound question. So at that time we couldn't, but now we know that these compounds are new. Okay, these are compounds are new, but we have to now see the library and extract that compound and then say do the NMR or other things. Okay, to identify that. So we couldn't identify at that time, but we know that these are the compounds. So we'll be doing that. Okay, we'll identify that. So now after that we'll see how this enters. Okay, how this this forms a pore in the fungal culture and therefore they enter. Okay, so there are some secretion systems. Okay, this bacteria is producing. Okay, so they are releasing in the media and they are producing some pores in the fungal culture, and they are killing. So how they are releasing? How this compound is being released? So this bacteria, when they are releasing this, how they are releasing? Okay. So there are lot many ways by which they release in the environment or in the fungal culture. Okay. So these are known as your secretion systems. Okay. Secretion system means they have secreted out these compounds to the outside world. Okay. Altogether, there are 9 types of secretion systems in the bacterial cell wall. Okay. How they will pass out to the environment or to the bacteria. Okay. So when we did the sequencing, we found out that this is type 4 secretion system. Type 6 secretion system, and this is like your injection type of system. Okay, so they will have this is suppose this is the bacterial cell wall. They will be having this type of syringe over there. Okay. So they will push this one in the media and they will go outside. Okay. This is a syringe type of thing. Okay. So they will push this compound outside. And when they come to say this is the fungal activity, this will go and they will punch it up. Then this compound will. Okay. So this is how they will create the pores. Okay. So there are a lot many secretion system, and we studied that secretion system, and that secretion system was involved in secreting of the compound. Okay. So okay, like that. So we did the transcriptome analysis. Okay, so we isolated the RNA and see the transcriptome and say some genes will be upregulated, some will be downregulated. Now suppose this is the bacteria, this is the fungus. So here, this is also work. Okay, so they are working. So we have extracted the metabolite. So when they are interacting, they will be producing some new compounds. Okay. So now this DNA is producing. They will be transcribed to some RNA and then proteins like that. So they will also do. So during this, some will be some new DNA will be activated to produce the protein molecule. Is it not? So at this time, what is being done? So some DNA sequences will be more more what do you say, more readily expressed. Okay? More readily expressed means they are upregulated. And some during this interaction will be downregulated. Means you don't work, you keep quiet. Okay? So some genes will be kept quiet. Okay? So they are only the downregulated, and some will be more active at that portion, and then only the upregulated. Okay? So downregulation and upregulation. So when we found the transcriptome, say we found out that some of the genes were upregulated. Okay, there are many. We are showing just this one. Okay, and some genes are downregulated during this interaction. Some genes are not working. Say they are silent. They need not to work. Okay, some need to work. So they are saying that. Okay, transcriptome is saying that. So we need to prove it. Okay, we need to prove it. So we will prove it by using some real-time PCR. Okay, real-time PCR क्या क्या सीन है? What is, how do we do this real-time PCR? Any idea? What is it? We have studied this or RT-PCR. How? Why this is known as a real-time PCR? Real time. Why? You will do that? Yeah, this expression they will be shown in real time. During COVID time also we did it, RT-PCR. So we can directly say this is this. We don't know. Is it not? So they can say directly. And it's very easy. Okay, or very accurate. First, we will require the PCR conditions and everything. So you will say that this is this one. One CTL was there. Yes, invite So, say, changing this value will say that this is this, this. Okay. So during COVID times also we did the RT-PCR, is it not? So that RT-PCR will be using, and then we say that these genes are working. Okay, so this is not very required. Okay, so these are some of the structures of your secretion system. I say that there are many different types of secretion systems. These are some of the structures of your secretion systems. Okay, these are some of the structures. So this is what we are looking at now. Okay, so we did the PCR primer designing also for your pigments and for your secretion system, and to do the real-time PCR. So quantitative PCR, and say we did that one. And then we can say that significant upregulation of murine lipoproteins were there, and TSSJ suggested that this is type 6 secretion system are involved. Okay, so this is involved, and producing biosynthesis remain unchanged during this interaction. So you can say that my vector is there, my fungus is there, they are producing this, say 1 kilogram per something like that. When they interacted, this is unchanged. This is not changed, they remain changed. So during interaction also producing is not being changed. Okay, so they are same. But other genes are upregulating or downregulating. Okay, so and then again we did another work which is fungal interaction. Okay, so fungus also say if they are alone they will work. If they come together, they will say talk to each other. Okay, they will try to kill each other or do something. Okay, so during this interaction, they also created some pores. Okay, what they did, they produces this brown color compound in that interacting zone. So see, from this side, one fungus is there, from this side, one fungus is there. So if they are same, they will mingle out. Okay, they will mingle out and then they will go. If they are not same, they will try to say do something. Say if they are say they can kill each other. Okay, so at that interacting zone, they will produce this brown colored pigment. Okay, this is your melanin. Okay, melanin that is present in your skin. They are good for your skin. Why? Because they can stop the UV rays. Okay, they are also involved in your pathogens. So in fungus, they are apart from your this UV rays, they are also involved in your pathogens. So they have been pathogenesis of this fungus also. Okay. Melanin. Okay. So they produces melanin during interaction. Okay, and we purified the melanin using say again TLC. This is how your cells look like. Okay, your hypha look like. So this is T coccinea. During the interactions, they will form some spiral. this one. Okay, so these hypha are this same, they are normal ways, but when they are interacting, they form some coil structures. By this way, you know that they are say interacting. Okay, some structural changes are there when they are trying to kill each other. Okay. This is one. Okay. And when you see through that electron microscope, you can see some pores here. Okay. Yeah, these are the pores. Okay, these are the pores in the hyphae. This is the normal one. Okay, these were the normal ones. You can see the normal hyphae, and this you can see the pores are there. So melanin, when they are produced, they are now filling the how these are creating some pores, and this can be shown through your scanning electron microscope. These pores you can see. Can you see? These pores are there. So they are filling the fungus. Okay. So again we check isolated the metabolite. Okay, and say again interacting on same what you say control bacteria and interacting bacteria. Again, some of the results like cinnamonic acid is produced during interaction and also in there alone, T-coccinase alone, and some are produced here. This alpha is produced during interaction zone. Okay, only in interaction zone. Tyrosine is produced during the interaction zone. Okay. Interaction. So these are the precursors of melanin. Okay, so to produce melanin, these things are required. So they are produced during the interaction zone. Okay, this melanin is there. Okay, melanin is produced during the interaction zone. So these are the precursors. These are the precursors. So cinnamonic acid is also one antibiotic. Okay, so you are finding this type of secondary metabolites. Okay, so again we purify the compound. And we extracted the brown compound. We know that this is melanin, so we do the UV spectra, then TLC analysis, do the solubility test. All we say it's a melanin. Okay, and then we need the FTI. So to know that compound, okay, so to know that compound, you need to do some biochemical analysis. FTI is one of the criteria. FTI is FTI का फुल फॉर्म क्या है? Yes, let's do it. Another one here. Fourier transform spectroscopy. So this way by you can know the compound. Okay, so we have purified this compound, isn't it? So we have purified this compound with TLC, and now we'll see the using FTI. And another technique is there, which is your NMR. NMR to KSL, what is NMR? Okay, so we know the compound, purified compound. Okay, what this compound is. So we have purified and we now check through FTR, NMR. This is this compound. Okay, so मुझे paper produce करना है. Okay, so that. Okay, so this is my lab. Probably you work with it today, isn't it? In the microbiology lab. You go outside. So this is Dibba, you have met him, isn't it? He is the man who did this work. And another girl who is not here, Samim, and the other guys are on there. So this is how you do. Say, suppose you have the problem, what techniques are you, what results you are seeking, okay? And you have to know the techniques. Okay, this is suppose I need to purify the compound. How will you purify the compound? Okay, I will purify by PLC or PLC like that, and then purify I need to identify. I will do the LCMS. I will do the LCMS. Okay, after LCMS, I will do the NMR, FTR, do the compound. Okay, so the people who are produce if I will do what I will do say gene expression studies. Okay, that will be some paper, good paper. Okay, so this is how you do the work. Okay, so there will come. I have to identify the bacteria. Okay. You cannot identify bacteria simply by using single word. Okay, single way. You have to use many methods, which is known as your polyphagic. Okay, polyphagic. So we have identified. Now we use the interaction. We split the compound. Okay. So these compounds are very important. This melanin is very important for anti-cancerous work. Okay. So they have been doing this. synaptic acid that we have. The other compounds. Okay, so they are very important. So by chance, which name is there, I will get Nobel Prize. And I will get $10 million. So by that thinking only I am working. So you should also work. Okay, you should also work. Something you are saying new thing, so easy. And then you can work. Okay, you can say your how you can work. Okay, so you can combine this work to other things also in plants also. Okay, so by that way also you can work and you can find say very good compound, okay, which can be very important for your research or for. Okay. Are you satisfied? Anything that you want to discuss? For extracting culture,
We use the half strength of Na and half strength of PPA.
okay, we use half strength of Na and half strength of P. But if you enable to know much, so we use the half strength. Or even try the video. We use the good. Anuma?
Nice and
Yes, you know.
offline not available.
Yeah, very good. Anuma? Anyone? Okay, so we cannot do that. We do not do that. Okay. One of my students, she was doing this. Okay. And because of this program, she could do that and we stop that program. But we can do it. Yes. Normal one. What is normal? Equal this produces we test. Because this one is not normal fish are producing is not normal. Means they are not producing very high amount of but for melanic it's okay. Kita hidup lagi, so practically kita hidup lagi production lagi, ini juga practical. So some of you are also doing community health, not absolutely different. So by you are like, so now here, anything? মানে আমি কি করলো দুটো আসলে
আমি দুটো সাহায্য করব
আমি তাতে জয়েন না because now we know that this is this compound, so this is what do you say, this is okay. We are not going to we didn't find any new ones, okay, or we didn't try to find new. So
This is called reference-based identification. So here we identified the peaks which were produced by this compound. Okay. So these peaks are almost similar to the previously reported already characterized compound. So that's why we have to go for reference-based analysis. When we have to identify the de novo compound analysis, then we have to go for some higher techniques, like MSMS, where the tandem mass spectroscopy, where the compound is broken down to small pieces. Then individual pieces are identified. Then we can compare it with database, and we can identify it. So based on those vector pattern, we can identify the novel compounds. But for already available structures, we can just we have to simply compare our spectra with already available.
Suppose there are no spectra available. There are not many compounds where there are no available information. Okay, for that you have to work. For that you have to work. You have to verify, then do the MSNS. Otherwise, and that will be a problem for us because we have to move from these and other places to do that. Okay, we don't have the facility to do that. Otherwise, this is all unknown. Unknown we can get.
Yeah, unknown for to characterize unknown probably you will need help from biochemists
Right.
so that they can know what the structure is, whether the double bond, single bond, those structural things they have to basically find it out. So if normal biotechnologists are done. Then those who are working on what is that MS, right? Mass spec, probably they know a little bit about how to characterize the structure or make the structure for the unknowns. Thank you, Rohit.
Thank you.
For I'm sure it they applauded so loudly that itself says that you have given a very good lecture. And he has a lot of secret research work going on which he doesn't want to share with his peers. One of them is me. So even online I could access his presentation. So now I know this structure and I have stolen it. I can take a picture and I have stolen it. So this is the structure of melanin. Okay, great.
Apart from that, we are also working on detection of some viruses by using again that strip that they use for COVID testing. That strip we have been making to detect some plant viruses, not the animal viruses, like CTV, cytosine virus, and that we are amplifying. So we have made that. So we have to make that.
So that's a very good contribution to the university basically. He's talking about making history. You can go to the field and you can just detect the virus easily. Using the district like they are doing for a COVID testing. You don't need to do the real-time PCR or anything. If you are in the field, take the district with you, just take a little of SAR, plant SAR, and then dip the district and that will give you information with the virus is present. That's it. Very, very easy technique. It's a very good contribution to our department as well as to the university. Thank you, Robin. Thank you so much. You have a practical session and equality is waiting for us. Tomorrow morning, so I will start at 8:30. Unfortunately, I will start at 8:40, 8:30. And then also tomorrow evening we had a validity section. So I want to represent to speak. We need the feedback from the people. What is feedback? We may not be able to improve in the near future. So you select representative and also you list out what you want to say. Because time is very short, we need to be able to give you 3 to 4 minutes to speak. Not all, but represent. From each university, you can select one representative and you can speak. We can speak not only on the lectures and the practical sessions, also about the food, the expectations, and everything that requires improvement. Okay? This will help us to find out where the problems are, so that in future when we open and we are planning because this time we have got 200 to 300 applications. So we are thinking of organizing short courses, maybe to 1 week or maybe a month courses. We have Robin we were discussing. There are many students they apply and they people calling me they say that you have not selected me, you have not selected me. So we want to do a course, a practical hands-on course where you will be able to hold the people and do it by yourself. Okay? That kind of because this one was very short for 1 week we cannot show you all the things. We will divide it and we will share. Okay? Thanks. Thank you, Robin.
Thank you. Thank you, guys.
Tomorrow morning, I'm not going to speak a lot about science, but I don't know what a little bit of science. Huh? Want to know what? Are we talking about the virus or we talk about technology? I'm also going to talk about public acceptance. What kind of representation are you about? What is your representation? Official form of analysis. And mobile phone, somebody left mobile phone. Who is this? Maybe somebody from the microbiology department. Maybe personal thank you. अभी क्या है? पहले आपसे पढ़े. अरे कितना विषय से पढ़े देखो.